Our next panelist is Dr. Diana Montes from the Universidad Tecnológica de Bolívar. She holds a bachelor degree in chemistry at Universidad de Cartagena and also holds a PhD in environmental toxicology at the Universidad de Cartagena too. She has plenty experience in pharmacology, toxicology, and molecular biology, especially in computational and analytical chemistry. Please, let's welcome Dr. Diana Montes with an enthusiastic applause, please.
Good morning, everyone. First, I want to thank the organizing committee for such a great course. The name of my presentation is In Silico Toxicology, Environmental Pollutants Interacting with Breast Cancer Proteins. So the purpose of this talk is to show you some bioinformatic approaches used in toxicology and in pharmacology as well, especially the database development, inverse virtual screening, virtual high throughput screening, through my work on breast cancer and the interaction of proteins and environmental pollutants. The main motivation to develop this work was, of course, the high prevalence of breast cancer in women worldwide. This disease is also very interesting for toxicologists, as the main risk factors are not generic but environmental and lifestyle-related factors, such as chemical exposure, alcoholism, obesity, among others, including the exposure to the endocrine disrupting chemicals, which are compounds that affect the hormonal system and that has been broadly associated with breast carcinoma. This is the outline of my presentation. We will start with an introduction, and then I will explain the database development of EDCs DataBank, which is the first database of endocrine disruptors with three-dimensional structures for computational toxicology research available online. The second part is the inverse virtual screening and the identification of new targets for bisphenol A, which is a pollutant widely distributed in the environment as well as in different plastics, including food containers. Virtual high throughput screening of urban endocrine disruptors targeting breast cancer proteins. And finally, we will have the conclusions and final remarks. So endocrine disruptors are a broad range of compounds that affect the endocrine system. However, their effects are not restricted to the deregulation processes controlled by hormones, but has been epidemiologically linked to a wide range of health conditions such as reproductive disorders, diabetes, neurodevelopmental responses, immune response, obesity, cardiovascular diseases, and different cancer types. This is very concerning as endocrine disruptors are virtually everywhere. We can find them in electronics, computers, textiles, as flame retardants, in plastics, in personal care products, in household products, industrial pollutants, and pesticides. They can be also found in natural products as phytoestrogens and mycoestrogens. Therefore, there is a growing international concern about the potential risk of those compounds in human health of general population. Breast cancer is a very heterogeneous disease. As you can see here, there are differences even between patients. However, this can be subclassified in different subtypes according to the immunohistochemical profile. In luminal A, which is the most common breast cancer subtype, luminal B, which is the most aggressive, HER positive, which have an overexpression of the epidermal growth factor 2, basal-like and normal breast-like, which are also considered as triple negative because of the lack of overexpression of the estrogen receptor, progesterone receptor, and the epidermal growth factor. Breast cancer shares some pathways with other cancers, such as those mediated by the receptor tyrosine kinase, which is deregulated to enable an accelerated metabolism, cell survival, and protein synthesis and cell growth, which are common characteristics in cancer cells. This disease is also very regulated by hormones, and it is very influenced by the hormonal microenvironment. So there are two important pathways in this disease, which are the estrogen receptor mediated and growth factor receptors mediated. The ligand-mediated activation of the estrogen receptor leads the activation of the kinase cascade, which results in two products, which are the phosphorylation of the estrogen receptor, making it more active at genomic level, and the progression of cell cycles and accelerated metabolism, which is characteristic of cancer cells. As I mentioned before, there is not a consensus list of proteins related with breast cancer or genes related with this disease or biomarkers for this disease. So I made text data mining to find those proteins or genes related with breast cancer using a great tool called Fable. And I found that around 8,000 proteins were related with breast cancer. And from those, only a very small portion were also related to endocrine disruptors, which estrogen receptor with the more number of reference with only 10 reference. This is expected as estrogen receptor is the main pathway of this disease. Other proteins also were found related to breast cancer and endocrine disruptors, but compared to the total, this is only a small portion. Therefore, more research was needed to find all the proteins in breast cancer that could be related with the epidemiological studies that establish a relationship between the endocrine disruptors and this disease. The hypothesis of this work is that proteins associated with breast cancer are molecular targets of endocrine disruptors, and the first objective was the construction of the endocrine disrupting chemicals database. The second one was identifying molecular targets for BPA in silico, and the third one was to predict breast cancer proteins with endocrine disruptors by virtual high throughput screening. And also, validation of these results were made using spectroscopic techniques in vitro. So the first part is the creation of the EDCs DataBank, which is the first database of endocrine disruptors with three-dimensional structures of these compounds for computational toxicology research, general population, and academia. The first thing I did was the internal library of the endocrine disruptors. So I took the list of TEDx and the EU list of endocrine disruptors, which use a selection criteria that these compounds are reported at least once as endocrine disruptors in the literature. Then I download each compound from PubChem, and I create a database of those compounds using InstantJChem. So now we got an SDF file with the whole molecules, the three-dimensional structures of the molecules, as well as some structural information of them. Simultaneously, we performed the initial design of the database. Here we can see some of the preliminary pages, the home page, the advanced search page, the data sheet, and the download page. This was made in PowerPoint. And then I evaluate the list of requirements and the stuff I wanted to do, and I start the web programming of the database using Notepad, HTML5, and CSS. The database was constructed in MySQL using jQuery and PHP to interconnect the database with the web interface. Here we can see the architecture of the Endocrine Disruptors chemical database. Most of the information is stored on the server. All the information is completely searchable and downloadable. It also has been hyperlinked to some external links for toxicological information, keywords, and all the data was previously curated in order to avoid errors. The users can enter to this database by simply writing its name on Google or by directly writing the address on the web servers. There are different ways to go through the information. One of those is by the source icons. For example, for general population, they can only click here if they want to know what endocrine disruptors could be found in cosmetics. And then they got a list of compounds of endocrine disruptors that could be found in those. If you click in one of these molecules, then you got the EDCs datasheet, which have all the information of the compound. So there are three different ways to search the information. The first one is through the search bar. The second one is the advanced search, and as well through the search icons. In each datasheet, we will find the name of the compound, synonyms, sources, identifiers, structural properties, for features, and also the 2D structure and the 3D structures, and the source icons, which are very useful for academic purposes and as well as for general population to recognize possible risk sources. It also contains a section with downloads in which the different molecules can be downloaded in different formats, such as SDF, MOL2, PDB, and PDB-QT. This database also have an interactive applet, which is JMOL Viewer. You can move the molecule and make some simple calculations on it. And also, these have some hyperlinks to important databases in toxicology, such as the Environmental Protection Agency of the US, Actor database, and some ToxNet databases as well. Here you can find evidence and the articles in which these compounds are reported as endocrine disruptors, the external links, and the keywords. This is the download page. As I mentioned before, all the compounds can be downloaded and all the information stored on it can be downloaded. Here you can download all the compounds at once in SDF format or zip format as well. And there is also some statistical information of the molecules stored on it. It also contains a contact us page in order to encourage people who use the database to make comments and suggestions to improve the database. This was an statistic analysis that was made only for examination of the data contained on it. Here we can find the chemical space of the compounds. Each point represents a compound, and here we can find the three principal components of each of the ACEs. Here we can see the distribution of the numbers of bond donors, numbers of bond acceptors, numbers of heavy atoms, as well as the molecular weights of the compounds. We can say that most of the compounds are low molecular weight compounds. This database was created to incentivize the studies in silico toxicology for endocrine disrupting chemicals, as these are a huge number of compounds and it's difficult to make in vitro or in vivo experiments. So now we have visitors from more than 100 countries, and this is great. So the conclusions of this part are that EDCs DataBank is the unique database with three-dimensional structure for endocrine disruptors for virtual screening. EDCs DataBank is suitable for virtual screening and other computational toxicology approaches, as the molecular structures are available in several formats. EDCs DataBank is a valuable repository to study the molecular basis of the interaction between these molecules and macromolecules involved in several pathologies. This database provides helpful information for research, academia, and general population. So the construction of databases is very nice because you can help people to do research with data and it makes more organized. So the second part is the identification of new targets for bisphenol A. This was made by an approach that is called inverse virtual screening, in which you have only one molecule and you have several proteins to test. This approach can be also used for the identification of protein targets for new drugs, for example. So the first part was a data mining analysis in order to find the proteins that could be evaluated here. As bisphenol A is pollutant widely distributed that affect not only the breast cancer risk, but other different pathologies. So I use different tools such as PubMed Remainders, Alibaba, Chiliwood, and HiveHub. The proteins were downloaded from Protein DataBank, and they were prepared using civil program package. All the molecules different from the macromolecule were removed, including water, native ligands, and all ions. Some features were fixed, for example, the site change, and they were optimized in the same program package. The structure of the bisphenol A was also optimized using Gaussian, using the density functional theory at B3 lib 631G level. The parameters for the docking analysis was established by using Autodoc tools, using a blind docking strategy in which the whole surface of the protein was included to consider all the possible interactions. The docking simulation was carried out by Autodoc Bina, which is an open source program, and this gives the position of the ligand in the binding site, and it also predicts an affinity score, which was used for ranking the compounds and determine those that form the best complexes. We also review the interactions between the protein and ligands on the binding sites by using ligand scout software. This software leads to see which are the contact residues that interact with the small molecule, and also leads to identify the type of interaction with them, such as hydrophobic interaction, hydrogen bonds, or aromatic ring interactions. A validation was also carried out by comparing the crystallographic structure of a complex and the resultant from the docking. As we can see here, the pose was predicted accurately, and also we got a good affinity score. This is the text mining graph generated. So from here we select those group of proteins to be evaluated. In this case, nuclear receptors, circadian rings proteins, and other proteins highly related with bisphenol A. After the docking studies, we found that the estrogen receptor-related gamma and some circadian clock proteins such as CLK1, CLK2, and CLK4 present high affinity scores for bisphenol A. This is interesting as these proteins are involved in the expressosome complex formation and regulation by the phosphorylation of certain arginine-rich proteins. Here we can see some of the interactions predicted. We can see that most of the interactions were hydrophobic with the presence of some few hydrogen bonds. As well, the affinity scores were high and were comparable to the one determined by crystallographic structures. And the bisphenol A, it's placed in deep pockets, which suggests a conformational change of the protein and could explain the high affinity of those compounds. This is the validation result. And this is the validation with experimental data. In this case, I correlate the arodombina affinity scores with normalized experimental data, and we found that R was 0.75, which is quite good. So the conclusions of this part is that BPA may interact with other targets different from estrogen receptor. BPA seem to interact with proteins involved in several high prevalence diseases. Dual specificity protein kinases show the best in silico affinity for bisphenol A. Validation showed that the in silico Dodkin protocol reproduced well the BPA pose on the crystallographic structure, and BPA toxicological effects could be explained through its interaction with key proteins in signaling pathways, as many of the proteins that I show you in the table are correlated with the diseases and health conditions that has been correlated through epidemiological studies. Here is the third part, which is high-throughput virtual screening of urban endocrine disruptor targeting breast cancer proteins. So this part generates a huge amount of data with more than 180 proteins and more than 300 molecules. So the first part was identify the proteins correlated with breast cancer in the scientific reports. For that, I used this gray tool that is called Fable, in which you can add a keyword and it shows you the genes and proteins in a non-redundant manner of those that are related with this term. Those that have more than 100 hits were taken for downloading the proteins from Protein DataBank, and the preparation and optimization was similar to the one I presented you before for the second part. The high-throughput virtual screening was carried out in an edge rack with Power 7 processors, and the docking process was carried out by Arodoc-Pino. One of the complexes was also chosen for interaction analysis and the validation of the interactions through protein ligand complex by spectroscopic methods. These compounds were categorized in several groups. One of those were dioxins. In this group, we saw that the complexes that exhibited affinity score lesser than -10, which is very, very good, were these two. So we can infer that endocrine disruptors could be affected the stress response, oxidative stress response. Here we can see the interactions with catalase and CYP1A2 proteins. In the case of plastics and other types of polymers, this is very interesting because the plasticizer that is more commonly studied is bisphenol A, but in this case I found that many analogs and substituents of bisphenol A also interact with breast cancer proteins and even with a higher affinity, which is very concerning, as when we go to the market we saw the label that says BPA free, but probably it contains some of these molecules that could be even worse. These proteins can interact as well with other proteins involved in several pathways, which make it more complicated. These are two proteins that are related with the energy production as well as the suppression or promotion of tumors. In the case of everyday products, we found that several endocrine disruptors were presented good affinity for proteins, though these endocrine disruptors are present in a wide variety of everyday products such as household products, cosmetics, drugs, antimicrobial, and personal care products. Here we can see some of the interactions. Most of them were hydrophobic in nature with the presence of some few hydrogen bonds. In the miscellaneous compounds, we found that those related with combustions were the ones that presented the higher affinity scores. This is interesting because many of these polyaromatic compounds has been related to carcinogenesis through a DNA damage mechanism. So they could be probably interacting as well in a different manner through protein ligand interactions. This is an interesting case with the protein CYP1A2 and benzo-A-pyrene, which is a non-carcinogenic compound. The results I show you here are just the top results, as there were huge amounts of data generated. Here you can see the heat maps that were made on the R program, which is a powerful statistical program that is as well an open source. The red ones are those that present the higher affinity scores. As you can see here, endocrine disruptors are as well very promiscuous compounds. So probably this could be the explanation why they can affect the human health through different health conditions and diseases. This was the protein I used for the in vitro validation. It is SHBG, which is a hormone transporter. So interestingly, the bisphenol A binds this protein in the same pocket as endogenous hormones. So they could compete by this place, increasing the free hormone levels on the blood, and this is also correlated with breast cancer. This is the workflow for the protein expression. The purification was carried out with a novel method called DynaBits, which leads to isolate the protein in a gentle way, keeping this in nature state. This uses copper nanoparticles that binds the his-targeted proteins, and this use as well some magnets to release the excedent and to keep the protein purified. The experiments to analyze if the protein was functional and to see the conformational changes after the binding with bisphenol A were microscale thermophoresis and circular dichroism. Microscale thermophoresis uses an excitation light, which makes the movements of the molecules give a signal. This movement is incentivized by a thermal gradient, and it changes with the interaction. So when the protein interacts with other, it generates dosage response curve like this. And the circular dichroism that measures the difference between right and left polarized and circular dichroid light. Here you can see some patterns of the results. Here in yellow you can see the typical response for alpha helix, in blue the typical response for beta sheaf, and in red the random coil. So you can, for example, establish if the protein is folded with this method, and you can also see if there was a conformational change by measuring the protein before and after the exposure to the chemical. this is the workflow for the recombinant plasmid design. The plasmid used was PET15MHL, as this had penicillin-resistant gene and also the his tag. The protein expression was correct, as you can see here, and you can see there is nothing in the controls. This is the recombinant protein after purification, and the molecular weight was in the expected place. Here we can see the results of the microscale thermophoresis, and we can see tendency to bind BPA around 1 micromolar. And these are the results of the circular dichroism spectra with the protein alone and the protein in cube at time 0 and overnight. Here you can see the results in a more friendly way. As you can see here, there is an increase in the alpha helix content of the protein after the exposure of BPA, which is a signal that the protein and ligand effectively interact. There was some statistical analysis as well in order to see if the results were statistically significant, and we can effectively see different in the conformational change after overnight exposure to the chemical. This means that they effectively bind, but also that the interaction could take some time to occur. So the conclusions in this part are that endocrine disruptors have the potential to target breast cancer proteins. Many endocrine disruptors are frequently inhibitors. I mean, there are promiscuous compounds that can bind several proteins, and the mechanism of actions may be very complex, affecting several pathways. The derivative and substituents of BPA presents high affinity for breast cancer proteins, and experimental validation was performed on testing the interaction between BPA and one of its predicted targets by circular decrease, and the main conformational change occurring was an increase on the alpha helix content. So here we can see that endocrines in disruptors could affect the signalings related to breast cancer. The data generated in this work could be useful to guide in vitro and in vivo experiments. This also was a good result of this research, that is BPA analogs tend to bind strongly breast cancer proteins, which could help to develop some regulatory laws in the future. And one of the contributions of this work was the creation of the database, which could be useful for making toxicology research by computer-aided methods. These were the publications of these results and some reference use. These are also some softwares that can be used for you if you want to make some text mining or virtual screenings. I want to acknowledge the ICGEB course, the Universidad Tecnológica de Bolívar, Universidad de Cartagena, University of Cambridge, as some of those experiments were carried out then, and Ciencias Bogotá. And this is a group of undergraduate students of the University Tecnológica of Bolívar that are working on bioinformatics and environmental research. Thank you.
Thank you all, Dr. Diana. Now it's time for questions and answers. So again, if you have an answer, just raise your hand and I will make the mic reach you. Does anybody has a question?
Thank you for such a great talk. As you were speaking, I'm sorry, I was on WhatsApp. No, I was not on WhatsApp. I was looking at a work that was published in 2007 by BioInformatics. What they did was that they created a human disease network. So, looking at this disease network, they link the breast cancer with colon cancer, lymphoma, gastric cancer, prostate cancer, something that I don't know what it is, Fanconi anemia, epilepsy, obesity, diabetes, and lots of other diseases. So the way that these diseases are linked are through, because they share some genes.
And probably some pathways as well.
Yeah, so my question is, have you tried to, since you have linked these EDCs to breast cancer, it is also possible that the same proteins that they are targeting are also related to other diseases?
Yeah, of course. And actually, one of the proteins we found was, for example, PKC theta, which is a protein found in many crosstalk signaling pathways. So, yeah.
But have you tried to approach to figure out which of the genes that are related to this cancer are also related to other diseases, so we can make the link between the diseases and different diseases?
Yeah, as I mentioned, there could be some genes shared, but there is also another proteins of these compounds, and it's the presence of aromatic rings, and they are also small, which are good properties for interact with proteins, as many proteins have in the binding site histidine and arginine-rich regions that could interact in great ways with these privileged moieties.
Is there any other
I have
question?
just a very simple question. Did you take a look at a similar number of proteins that are not known to be involved in breast cancer?
With endocrine disruptors, or
just plain old randomly chosen proteins? I mean, in other words, did you show enrichment for PPA interaction in your breast cancer set relative to a background that's outside of breast cancer?
yeah. Actually, I select some group of proteins involved in important processes in the cell, like cell growth and circadian rhythms, for example, and breast cancer, and which I found that is that they interact well with these proteins as well, and especially to those that are related with the same that are related with the same health conditions that were predicted by the epidemiologically research. So,
Is there any other question? No? Okay, so thank you again, Dr. Diana.